Esteemed candidate, the College of Deans of this university, represented by us, have read your thesis and the propositions that go with this, and is ready to hear you to defend it. Please take a seat. Does anyone in the audience wish to exchange ideas with the PhD candidate? If not, I would like to ask the first proponent, Professor Luc Hartman, to start the opposition.
Dear candidate, I enjoyed reading your thesis. It's very comprehensive, and I particularly like that it could have some potential to be used for more things of the same nature in the future. I think it has good potential for growth. I want to start out by asking you a few questions about chapters 5 and 6. Mm-hmm. Starting with chapter 5, which uses ABC to analyze diversification rates, and it's one of the first times that type of approach has been used before. So I wanted to explore your thoughts on how well it works. In particular, one of the things that you have to do in an ABC analysis is choose a set of test statistics. And the test statistics that you choose have a large effect, as you discussed in that chapter, on your results. One of the things I was wondering about when reading that chapter is about other types of test statistics that could be used, and whether you think that they would be informative or not for the model that you used or for other models. And in particular, the kind of test statistics I'm thinking of have to do with tree balance. So can you just maybe share your thoughts on tree balance metrics as a test statistic, whether they would make your method work better or not, and then what types of models do you think they would be informative about?
Highly learned opponent, thank you very much for your question. So to use tree balance is obviously a very informative statistic, especially if you have unbalanced trees. But the specific models that we looked at in this chapter are all balanced, so they don't necessarily generate unbalanced trees. So on average, you would find that all the trees have the same balance. So then there's not much information carried by such a summary statistic. So that, I think, wouldn't work. But you could imagine other models for which tree balance would show some variation across parameter values. And then I think tree balance is certainly a very good statistic, especially to supplement the normalized LTT statistic, because that completely ignores any balance or any topology features at all. It only looks at branching times.
Right. Okay, good. Can you talk a little bit about one of the, in phylogeny building nowadays, there's a lot of discussion about gene tree, species tree methods, where one makes a set of gene trees and then applies, say, coalescent theory to build them into a species tree. And typically in comparative methods, we don't have that type of approach leading into our analyses. Do you view that as a problem? What kind of effect might that have on our conclusions?
So, typically we generally take the best tree that we can find, and we assume that it's some kind of reflection of the species tree, and we go from there. So, in that way, you ignore all the noise and fuzzy stuff that comes from the alignments and aligning all the stuff and the substitution models and all this background complications that come into generating your species tree in the end. So future models maybe would be better suited to directly work on these alignments. And that would certainly be a great step forward as well, to do diversification analysis on alignments instead of the tree itself. But given that, so I'm not completely sure, but I think that already creating such a tree, so that's what these molecular people do, takes them a lot of time. And there's a lot of considerations that you have to take into account, and also constructing tree itself is quite a time-consuming process. So including that as well in our analyses it definitely doubles our work.
So do you think that the gene tree, species tree types of approaches are just another example of more elaborate phylogenetic construction methods, and there's no particular biases that we might face from using trees that ignore that distinction between a gene tree and a species tree?
No. So if you ignore all these, or don't really look at all the assumptions that are made during the creation of such a tree or the reconstruction of such a tree, then you're, the idea is that you then can sometimes such choices could potentially influence later made inference using that tree,
Right, okay.
as we also show in chapter 4.
Yeah, great. So, okay, so let's talk a little bit about next steps that you think would be a good direction to go in following up on these chapters. So in chapter 6, you use your approach to fit a model of sympatric and allopatric speciation in cichlids. And I think it's a good example of taking comparative methods from where we are right now, which is using simple models with relatively few parameters that are kind of loosely connected to biology, to making them better connected to the biology of organisms. Can you talk about, if you were to do that again in a different system, what's an example of a model that you might be interested in testing?
In a different system?
Yep.
So, I think, I wouldn't really know. So, well, obviously, in the current model, there's this very classic dichotomy between allopatric and sympatric speciation. And, well, we know that basically both types of speciation don't really exist. It's more or less everything is in the middle. So we should need to come up with a slightly different way of implementing these effects of these environmental changes on the diversification, if there is such an effect. And then, since we already saw from our analyses that we often find the exact same results that you would get when you would just apply the prior that the people used to generate this tree, we would come back to your previous question again. So then it would be interesting to not only track the inheritance structure, so how the species relate to each other, but also how their genome underlying that changes. So you would also take into account substitution rates and clock rates and all that stuff.
Right, yeah. Do you imagine
it would be a much more elaborate model then.
Yeah, so would you want to add some new test statistics that would capture that kind of thing, or would you just use a different simulation model, but basically the same approach?
I would use a different simulation model, but the approach would be the same.
Yeah, okay. So there's been a few papers recently. So I understand, and I'm on your side about pushing comparative methods towards more elaborate things with more parameters. But there also has been a little bit of criticism of that kind of approach lately. So if you think of Dan Roboski's paper saying that we can't estimate extinction from phylogenetic trees, for example, or I don't know if you saw Emma Goldberg and Dan Roboski's paper critical of the BISI approach, basically saying that you can get too many false positives with complicated comparative methods. So do you have a response to that kind of criticism that these models are sometimes very difficult to fit or have bizarre statistical characteristics?
Well, as I also say in one of my propositions, one of the biggest problems with applying all these models is that they're all usually internally quite coherent. So data generated by the model, if you fit the model on that, you get the right parameters again. But there's no way to check whether, if you have a real phylogeny, which model best applies. So unless you would have a phylogeny for which you would know which species relate to which went extinct when, which is inherently problematic because, well, we need data for the past few million years for that for certain taxa. So we don't really have that. I think there's a number of true phylogenies for which we do know stuff, but that's mainly on bacteria and mice strains, I think. It's work in the 90s. So apart from that, it's really hard to get some good feeling about whether these models represent something that really happens or whether they describe some way to describe the data best. So that ties nicely with your talk yesterday about this adequacy stuff.
Yeah, okay.
So we have no clue. Right.
Do you think that there are some hints that you can get about certain type, like how do you know if you're considering a hypothesis whether it's testable or not before you?
Well, so obviously it's not completely useless to do this stuff. So when you apply such a model, then you also generate new ideas about how things should have worked, and you can maybe test those. What I really like, for instance, is the diversity-dependent model of Rampal. So there you could kind of reconstruct the past diversity of a certain clade, and then they were able to check that actually with the fossil record of foraminifers. So in that way you have kind of an independent double check of your findings. because you're not directly fitting on the diversity that happened back then, but you're just using your molecular data in order to tell something, to infer something about this diversity. And then it turns out that when you compare that with the fossil data, it matches.
Right.
So that's kind of super awesome.
Yeah, okay. And what's your philosophy about connecting the statistical results of your methods to biological conclusions? Sometimes it's that step can be more or less difficult, I guess, depending on the approach you're doing. And I know that this question is a bit vague, but I'd just like to hear your thoughts.
So you mean like, so if my model, so there's some outcome of my model and I see some kind of fit of my model?
Yeah,
To which
so,
extent
so well,
am I
there's various
describing-
things you could look at. You could look at the parameter estimates themselves, you could look at their confidence intervals, you could look at overall model fit. But the goal here is to do biology, so
Yeah.
what's your approach there?
So initially you try to focus on finding those parameters for which you explain the data best. So you try to find that model version that has the best fit. But obviously you don't know whether that's a really good description of your data. So then you would have to go back to your parameter estimates and see whether they make sense at all, or whether they're completely off the scale somewhere. And I think, so generally what I have done, I've not really, so when all the estimates that my models give, I've mainly one-on-one translated them to biology, I think, in all my chapters. But especially in the last chapter, I think we have tried to look at very many different ways of the versions of the model in order to find whether these changes in water level, for instance, really are that important or not. And then it turns out that this is not so easy to determine, especially when your inference method tends to, when the information in data is not so high as you would want. So especially when we started to include more and more parameters, then it turned out that it became harder and harder to estimate these parameters correctly, or at least the uncertainty around these parameters started to increase. And yeah, then you start running into problems also for interpretability.
Right, okay. Do I have more time, or am I?
No, I'm sorry.
Okay.
We have to continue now. Thank you very much. And I would like now to call upon Professor Bokma to continue the opposition.
So dear candidate, I really enjoyed reading your thesis, especially as it provides a mix of empirical, theoretical, conceptual work. I would like to start by exchanging some ideas about proposition number 2. So I would like to ask the paranymphs to read aloud for the audience the proposition number 2.
Diversification rates should not be estimated based on phylogenetic trees which were constructed with other aims in mind than estimating diversification rates.
Thank you. So I would like to start by asking you, what do you think is the purpose of most phylogenetic reconstructions?
Highly learned opponent. Thank you very much for your question. So I would say that most phylogenies are constructed in order to understand which, so many of them are with, so it depends a bit on what kind of taxa you're looking at. So if you're looking at higher taxa, they're often also used to find whether certain species descriptions are correct, so to do taxonomy basically. And I guess when you're going to more lower taxa in which that's less of importance, then you could still look at whether large groups, how they are related to each other and how they relate to each other back in time, basically.
Would you agree with me if we could generalize that by saying that many phylogenetic trees are reconstructed with the purpose of reconstructing natural history as accurately as possible?
Yeah.
So in that case, do you think those trees would be fit to use as a substrate for estimating diversification rates?
If they would be accurate, yes.
But if the purpose was to make them as accurate as possible, isn't that then the best substrate for basing our estimates of diversification rates on?
That way. It's a trap. Yes.
Then my next question would be, how does your proposition number 2 relate to the title of chapter number 4?
Mm-hmm.
So chapter number 4 starts on page number 107, where you conclude that joint inference of phylogeny and diversification rates from molecular sequences does not outperform a two-step approach. I wonder whether you could philosophize on how that relates to your proposition number 2.
Yeah, so the idea, or from the outset of, so basically what I also say in proposition number 2, so we had the idea that when you construct a phylogenetic tree without just trying to capture the evolutionary history, then sometimes you might make assumptions that could bias further estimates using diversification analysis. And in this chapter, we try to test what would happen if you would jointly infer these diversification rates with the topology of the tree. We found that there was no difference. However, when we started to vary the clock rate model, if I'm not mistaken, the branch rate model, sorry, then we did find that when the variation in the branch rate model increased, that we could still found some examples of differences between these two approaches where indeed the model used in the reconstruction of the tree would bias for later estimates, or at least would introduce some kind of difference.
Very well. If I have time, I would like to come back to that issue, but first I would like to ask you some questions about proposition number 3. So might I ask Paranym again to read aloud proposition number 3?
For all practical purposes, approximate Bayesian computation needs not be approximate at all.
Thank you. So my first question would be, why is approximate Bayesian computation called approximate?
So normally when you do Bayesian computation, you try to obtain the posterior distribution of your parameters. And in this case, we're not using the exact likelihood, so we're not targeting the exact posterior distribution, but rather we're using the difference in the observed data and the simulated data, either directly or using some kind of lower dimensional summary statistic. And then where you would normally only accept those parameter values that would have a favorable likelihood, now you would accept any parameter values for which the summary statistic or the data is sufficiently similar, but not exactly, because it's typically, especially when your data gets a bit more complex, it tends to be very hard to get exactly the same data, so you try to find approximately the same data, with the idea that as your data becomes more and more similar, you more and more start to approach your true posterior distribution. So basically what you obtain is an approximate posterior distribution.
So using this approach, in what case would your posterior not be approximate, but would be exact Bayesian?
So the propositions is not necessarily in the mathematical sense that they are extremely exact, that they are exactly identical anymore. So that would be when your threshold goes to 0 in the approximate case. But that, for all practical purposes, that's very tedious to let the threshold go to 0.
I would like to get that point. So in theory, it would be possible that approximate Bayesian computation is not particularly approximate, because if your data exactly matches the observed data, then you get the exact posterior.
Yeah.
Right? Now, you just mentioned that in practice, that is not feasible.
Yeah.
But doesn't that precisely contradict your proposition number 3, that for all practical purposes, it does not need to be approximate?
Does not need to be. So I would say that, so as you can see also in, for instance, chapter 5, where we test all these different summary statistics and test these approximate Bayesian approaches, then we see that actually using an approximate Bayesian approach works as well as using the likelihood. So for the practical purpose that we're using it there, the performance is equally good as using the likelihood approach. So then, for all practical purposes, it performs equally well.
Very well. I would like to ask a little more about approximate Bayesian computation. So in several of the aspects of your thesis, you use approximate Bayesian computation to estimate the parameters of a particular model. Now, when it comes to, for example, estimates of diversification rates, we often do not know the model beforehand. I would like you to reflect on the possibilities to use approximate Bayesian computation to compare between different competing models.
Yeah, that's okay. So that's always tricky when you start to, especially when the models are not nested, then it becomes tricky to compare them, as both models might generate data that's extremely similar. So then you are stuck with, well, two equally well explaining models. But I guess that's, well, in the likelihood case, you also have that problem, I would say. It's not exclusively a problem for approximate Bayesian computation.
If you would have the likelihood, how would you go about to distinguish between competing models, do you?
Well, then you would use the A or BIC.
Yeah, base
If they are
base
nested.
vectors.
Or base vectors, yeah. You can also
Do you
do
think it would be possible to compute base vectors using approximate Bayesian computation?
that. I think... So I know that there is some papers out there that do use approximate Bayesian computation in association with model selection. But I've never really done that myself, so I wouldn't really know how to do that. It's not exactly straightforward, as you would somehow need to use probably the posterior distribution of your summary statistics and then compare the spread of that or something like that. I'm not completely sure.
But, would you agree that it would be an interesting set for your models if you could make comparisons between density dependent or pure birth?
Well, definitely, because then you would be able to, well, at least get closer to what would better describe your data.
Okay.
Although we both know that both models probably are not entirely realistic, but...
Thank you. Thank you.
Thank you. The opposition will be continued by Professor Witt.
Let me first congratulate you with your very, very nice thesis. I enjoyed reading it. I do have a couple of questions, and I'm sitting here as a statistician, so I'm going to ask you sometimes a couple of technical questions. So hopefully you can help me out a little bit. We had a nice introduction already about approximate base, so let me start with that one. In your in chapter 5, you actually are doing some of the you're looking at approximate base, and you propose basically this normalized lineages through time kind of statistic to basically use in your approximate base computations. Mm-hmm. Now, it's probably important to know that obviously there you normalize and you take away time, and effectively you take away the number of lineages in some sense. You just normalize everything to one. Now, when you take away any reference to time, I find it kind of interesting and surprising that somehow you're still able to estimate rates very well. So can you give me an idea why should you be able to estimate rates well, even though any reference to time has been taken out of your summary statistic?
Highly learned opponent, thank you very much for your question. So yeah, that initially also surprised me. In the sense that indeed so this whole normalization stuff we do because then it's much easier to use the summary statistic in an ABC context, because then your differences tend to be within a certain range. But from some pilot runs that I did, it tends to be that the shape of the curve changes as your rate changes. So if you have a higher rate, then I think the exponential curve gets pulled down. The normalized LTT curve moves more towards the right-hand corner. So that basically the massive increase is pulled more towards the present.
But
and if your rates are very low, then you get a more like a straight line.
But if your death rate and your birth rates are kind of going up simultaneously, will that still be the case? Will you still see a significant change? I mean, I can imagine that you might see differences in the variance of this curve. Mm-hmm. But that's very, well, there's not much information in variance, really. So I was kind of wondering, but apparently you do, you say.
As far as I can remember, you do see differences in shape, but I can, so you're referring to the problem of jointly inferring speciation and extinction rates, which...
Well, I mean, there's obviously all the three models have different, some of them have parameters. Some of them you can clearly see one might be able to estimate from this statistic. Others I was just a bit more surprised. And some of your chapters have very nice appendices, others, in this case, don't. So I was kind of interested to see some of the details, or hear some of the details from you.
Yeah, so apart from that, I've looked at these pilot runs. I'm not completely sure. So I can indeed imagine that. So normally when you try to jointly infer the birth rate and the extinction rate, you start to run into troubles, especially when they both increase in the same way. So then you start to infer diversification rates instead of the actual rates themselves. And surprisingly, we didn't need to do that here. And I don't exactly recall why we didn't need to do that here.
Okay. Maybe just a follow-up question.
Well, and I noticed that even the likelihood performs well, so that's surprising.
Mm-hmm. Follow-up question, maybe just a little bit. So obviously in this kind of case you assume you have full access to your phylogeny, even the died out branches, probably in order to do any of the calculations of your normalized LTT statistic. But in case you do not have it, like in practice, you don't actually have access to your died out branches most likely. So how would you change the definition of your statistic in order to still be able to match?
No, but we didn't include extinct lineages in.
So you took them out anyway?
Yeah. They were true, well, like phylogenies as you would see them in literature.
Okay,
like
so you...
reconstructed phylogenies.
Right, okay. Going back to chapter 4, maybe you can have a look at page 118, figure 4.5d. Let me just, while you're trying to find this, in this particular chapter you try to make a point that two-step inference in some sense is fine. Now, I personally can interpret fine in at least 2 ways. Let me just say 2. How do you interpret fine?
For all practical purposes, it works.
Well, I'm a statistician. I think of fine in terms of bias and variance. So, what do you mean?
I mean that there's no bias. Yeah.
Mm-hmm.
And the variance is more or less the same. Yeah, I don't recall exactly. But I think the bias is more or less similar between the two approaches.
Indeed. So I think you interpret indeed you're fine in terms of bias. I agree. I think that's what you do. I mean, in very general, basically, if you have a model and you do two-step inference, as long as the model is not completely non-linear in some manifold, you would expect that the two-step method would still be unbiased. But the variance could be quite different. Now, you do say indeed that you might have different, well, your variances are similar too, and you probably refer to the figure that I'm actually referring to you, or I've already referred you to before. So maybe have a look at that particular figure. In that figure, indeed, you see you have your method, the two-step method, and the joint inference method right next to each other. And in fact, it looks like none of them do very well. Is it surprising?
Not really, I guess.
No, it shouldn't be, but why not?
This has been some time ago.
So, okay, just, I mean, obviously, I don't expect you to remember every figure, but here you're trying to estimate an extinction rate, which is 0, by means of lots of simulations where you try to simulate from different things. And so,
yeah, so we're generally, this is what I referred to earlier, that we're overestimating this extinction rate. But we're actually diversification rate, so that's the figure A,
yep.
That works.
Yes.
Kind of.
Yeah, it kind of works, but obviously you actually know a priori that the other one is not going to work, because if you try to estimate something that's always positive, your estimate will always be positive, and so your 95% confidence interval, or what you suggest are 95% confidence intervals, I think, credible intervals, maybe you want to call them, are always going to be positive anyway. So we'll never include a true parameter. So when you say variance, actually here it's even an example of bias basically as well. So I think you, in your chapter 4, when you say bias is so your fine is in terms of bias, your bias is okay, or at least it's similar to any other method, but I don't think your variance necessarily has to be the case, that they are similar as well. Let's see, do we have time for another question?
We have a little bit of time.
A little bit of time, so let's just ask a very small question then. So maybe so chapter 3 you have you're working on basically trying to estimate dispersal ability in 2 taxi, or 2, you call them guilds, I suppose, and you define this parameter alpha, which is your dispersal ability. But you don't say anything about what value it can have. According to you, what values can your dispersal ability have?
Anything between 0 and 1.
Anything between 0 and 1. Okay, that's what I assumed as well.
Yeah.
But, you know.
Yeah, we don't specify that, no.
Then I'll leave it. Thank you.
Okay, thank you very much. I would now like to ask Dr. Roosendaal to ask his questions.
Dear candidate, thank you very much for my copy of your thesis. I really enjoyed reading it, and I think it's a substantial contribution to theoretical ecology and evolution. I have some questions for you. So firstly, from the introduction, you talk about phenomenological models and mechanistic models. So how would you describe a phenomenological model and a mechanistic model and the difference between them?
Very learned opponent, thank you very much for your question. So with a phenomenological model, I mean, with that I would allude to models such as the constant rate birth death model or the time-dependent speciation rate model, where we don't really know why these rates are what they are, but rather we use them to describe how the dynamics in the certain system that we're looking at work. So basically they're not really helping us that much, if only to see how things would work if these assumptions are true. Now, a more mechanistic way to go would be to actually include some kind of processes that would lead to changes in these diversification rates. For instance, as in chapter 6, where we take into account these changes in the environment, or if you would look at already a bit more mechanistic, but still quite phenomenological, the diversity-dependent diversification model, where we link these, where Rampal linked these diversification rates to the actual diversity around such a species.
Thank you. So on your step-cams model, is that a phenomenological or a mechanistic model, would you say?
I would say that's mechanistic.
Why is that?
Because I think we at least we try to incorporate many of the different processes that could work when you have when you look at species from a trait perspective. So rather than just describing how the community changes, we rather try to, from the bottom up, try to understand how these processes would influence the species composition of a community. So I would say that's mechanistic.
Great, thank you. So still on the step-cams model, you construct a community from all possible species that could occur in your area by starting with all possible species and then removing from them. Could you imagine a version of this where you start from nothing and build up, and would you expect to get the same answer then? Should you get the same answer?
So I think that would work more or less the same way, depending on how you apply your rules, of course. But if you would start from nothing and then simply would, so if you have a lot of limiting similarity, the first species that you add would be the one that experiences least limiting similarity. Or you have a habitat filtering step, so you first add the species that experiences least habitat filtering. Or if you have a dispersal assembly step, you add the most abundant species. So that inherently that would yield you exactly with the same results. It should. We didn't test that though. It might be faster as well.
Yeah, it might be indeed.
It probably is.
So thank you for that. So just moving on to chapter 3 now. So you included 2 different guilds of dispersal ability in these trees. So I'd like to know, how do you find that these 2 guilds coexist? Is there an advantage or a disadvantage to being in one particular guild, and is there a trade-off? How could that be incorporated into your model?
So this model is a bit more phenomenological in that sense, that we just decide, well, we looked at the data and we said, well, you have these that disperse this way, these that disperse that way, and we just make this cut between these 2 groups of species. I wouldn't really know whether why these species use different methods. I don't really know. I guess I could imagine though that those biotically dispersing species, they need to invest a bit more in generating fruits or something like that. I don't think they're all using fruits, but the majority might utilize the formation of fruits, which is then generally costly, but the benefit is that they get transported a bit further. And on the other hand, you have the abiotically dispersed species that don't necessarily need to produce these fruits, but maybe only very dry flotation structures. But the downside of that is that they don't get that far. So there will be a trade-off.
Okay, thank
you.
you.
It's not in the model.
Do I have time for one more question?
You have time for one short question.
Okay, thank you. A quick question then, please, on Proposition 6. So can I ask the Paranym, please, to read Proposition 6 to the audience?
City fish communities are just like savanna trees.
Thank you. So my question relating to this is, which savanna trees species do you know which mouth brood their offspring?
Let me think. No. I don't think I meant it in that sense.
In what sense did you mean it?
So I meant it from this, if you look at this trait-based community perspective, then we see in both these communities that actually the majority of this species composition is not so much due to these trait-based things, but rather tends to be very stochastic, or the background of these compositions tends to mainly be driven by stochastic effects. And surprisingly, that's both in savanna trees and in very different type of organism fish, which actually move around and do all kinds of other interesting stuff. And still, they are mainly driven by stochastic effects.
That's fantastic. Thanks very much.
Thank you very much, Lou. The opposition will be continued by Dr. Ma.
Thank you. Dear candidates, congratulations on this achievement. I enjoyed reading the thesis and also I learned a lot. And of course, as you know, this is very relevant to my work, but I am an empirical biologist working with cichlid fish in practice. And so I don't think about reconstructing models on a day-to-day basis, even though it's very interesting and relevant. So, my first question to you has to do with the relationship between speciation and community assembly. And am I right that you assume that speciation doesn't play a role in community assembly?
Very learned opponent. Thank you very much for your question. So are you referring to the assembly in the, so do you mean in the first two chapters or in chapter 3? So that's, well, so we
First
kind
two
of,
chapters, I guess.
so in the first two chapters we look at much shorter timescales in which there's not necessarily evolution, so there's only species replacing each other, but they don't really evolutionarily adapt. And in the third chapter we explicitly look at the dynamic equilibrium between these different migration types and also speciation in the meta-community. So there you do have speciation.
Okay, so let's focus on the cichlids, because I'm most familiar with those. So in those models, you assume that you sort of ignore that speciation would happen and have any effect. But then in the end, you conclude from your patterns that you find how cichlids have speciated. In other words, you use community assembly processes to make inferences about speciation processes. And that seems inconsistent to me.
I can see what you mean. Indeed, so in the end, I think I go a bit into speculative mode and suggest that since we don't really find these strong effects of limiting similarity or the environmental factors, that somehow this diversity in these cichlids is, so apparently this diversity in the cichlids is mainly driven by stochastic effects, which is very odd if you always think that these cichlids are so extremely diverse because they have adapted to so many different things. So that's how I then started to speculate that maybe then this diversity or this speciation in these cichlids has been driven very differently. But I can see that maybe using ecological data to extrapolate towards an evolutionary timescale might not always work.
Okay, thank you. And then my other question is about the title of your thesis. So, as you already discussed here, also in response to Professor Harman's questions, there is this problem of testing whether if a model fits the data, the processes that you included in your model really are the right explanation for those data. And then in the synthesis, you speculate that you can use, you basically need independent data for the final check, right? And then you talk a little bit about how you would do this in a phylogenetic context. You talk about fossils or making your own phylogeny with rapidly reproducing organisms. But how would such data look like for the community assembly problem?
That's a good question. So yeah, the idea would be then that so when you match these processes, then the idea was that you could, if you use the right model, then you would also generate new predictions or new stuff that you could measure in the fields in order to confirm that it's actually these processes, and it would be kind of independent measurements or something else that's not necessarily correlated to the data that you've already used to fit your model. So in these StepCam models, I'm actually not completely sure what kind of predictions it makes. Because I was also realizing when I wrote that that means that every model that I've made must have generated some kind of predictions that we should test. So I was hoping that I didn't get this question, but that didn't work out. So I think for the StepCam models, we could maybe, well, one of the things that I would expect is if you change the meta community, as we also like to some extent did in chapter 2, you would probably find different estimates for your different processes. But that's not necessarily a very independent way of checking your assumptions, I would say.
But what
You could also go out in field and test whether truly, if you put two cichlids that are very similar in trait-wise, you could then try and see whether they would undergo a lot of competition or start eating the same food particles or stuff.
Yeah, and what about community assembly experiments?
So I don't know any community assembly experiments.
That's why I use the word speculate.
Yeah. So yeah, you would allude to just starting out with some kind of combination of species, then put them somewhere in a pond and see what happens. Just wait. That could happen. That could work, I guess. It would especially be interesting if you would combine species that are, for instance, very similar, or you would introduce them into a habitat that they're extremely fit or unfit for, or mixtures of that. I guess that could work. I'm not completely sure, so that could confirm to some extent that these effects do exist. It still doesn't really tell you whether they are really important in the field, of course. But then that's where you can use the model, maybe.
Okay, thank you. I think I have to stop here.
It is time. Thank you very much. I would now like to call upon the last opponent, Professor Olf.
Yes. Thank you. Dear candidate, I've also read your thesis with great interest. It's a fantastic, I think, combination of deep theoretical work and also your own field work, and that's rare in this branch of work. I would like to ask you some things on page 153, chapter 6. So it's on the problem of Lake Tanganyika that splits in time. Every now and then it splits in two lakes, and then it merges, and it splits, it merges, and that's an important phenomenon you're studying here. And you show, and you indicate underwater level changes that there's 6 times in the last 500,000 years, or actually 4 times in the last 500,000 years, where the lake splits because the water level is 300 meters lower. What do we know about these periods? Why did that happen?
Highly learned opponent, thank you very much for your question. To be honest, I'm not too familiar with the African geological literature, but I think what generally happens in these periods is that there's periods of drought, or there's rerouting of the rivers such that they don't really flow into the lake anymore.
And what causes a drought that leads to a lake drop of 300 meters?
Wow.
It's actually not that difficult. So I have a graph here that shows you, if one of the pair nymphs can show it to you. It's actually called an ice age.
Yes, but
I know. So maybe you know about them. So if you first look at this graph, then you see that actually there's a perfect correspondence between your lake low periods and ice ages.
That's
so
nice.
That's helpful, right?
I didn't know that. Yeah.
But now look at the other side. That gives actually the climate over the last 5 million years, and that's your period of interest, right? So, as you probably know or should know, that ice ages are restricted to the Pleistocene, which is the last 2 million years. From 2.5 million years until 5 million years ago, how many ice ages were there? Can you read from this graph?
So I'm not an expert in reading these graphs, but I would suggest not so many.
Like how many?
So if these blue parts are glazed, so I guess around 0.
Yes. So that's called none, yeah. So in the half of the whole period you're interested in, there were no ice ages. And the ice age is actually a pretty close predictor of your lake level lows, right? So
it appears so that there's a strong correlation.
Yeah. So instead of going to great trouble in predicting actually when your lake was low, and because you say I have no information on what happened between 1 million years ago and 5 million years ago, and you make all kind of assumptions and speculations, like let's say that ice ages happened at random. You could have looked a bit in the geological literature to see actually what's causing these low levels, right? Is that a suggestion?
Yeah, that's a good idea. So then, but that's also, so then this would still, so this would mean that the first about 3 million years, you would only have diversification through, like in a relatively high water, high lake level environment, and from then on you would start to have this accelerated effect due to a number of...
And probably not more than 10 times. Yeah.
Probably not.
Yeah. Well, but because that is good. In the last 1 million year, you actually have the geological information from the lake, and that's also the period when these ice ages happen. So that's actually nice. But beyond that, actually, you don't need to be very concerned about lake level lows because simply there were no ice ages. So that's
but on the other hand,
The College of Deans will withdraw for some brief period to consider its decision. U kunt ook gaan zitten. Esteemed candidate, the College of Deans of this university represented by us, having read your thesis and the accompanying propositions, and after hearing you defend it, has decided to award you the PhD degree of this University of Groningen that you so desire. Your first primary supervisor, Professor Etienne Rampal, is authorized to award you this honor. Please.
I'm happy to accept the task assigned to me by Director Magnificus. However, before performing this task, I would like to address a few personal words to you. Dear Thijs, in 2010, I was struggling with a manuscript that had been dragging on for years and years. This manuscript dealt with the question whether species with small body sizes diversify faster or not. Our data was inevitably biased towards vertebrates, so we had to make a phylogenetic correction. Originally, I had done this using classical taxonomy, but a reviewer insisted that we use more modern methods. He was asking for nothing less than a tree that spanned from midges to elephants and claimed that this could be done in an afternoon. I was rather desperate, but fortunately there was you. I'd originally hired you for some other task, but you were exactly the right person for this job, because it needed to be done quick and dirty. Although it took a few weeks, you produced the required tree, and the paper was eventually accepted in Interface Focus, thanks to the contribution of 4 other people present today, one of them as very critical reviewer. This role of project rescuer suited you fine. Likewise, you assisted Fons van der Plas in the project that led to one of their chapters of your thesis, and your parent in Jasper in finishing one of his chapters. And I've recently asked you to help out another PhD student of mine, Adriana, with one of her manuscripts, which we just even discussed this morning. Pleased by the aforementioned accomplishments, I offered you a PhD position. But despite the Dutch word for PhD student, a PhD student is more than an assistant. So it's, in Dutch it's an assistant, but a PhD student PhD trajectory should really result in an independent researcher who can lead his own projects. This was maybe less obvious for you. Partly, this was due to the fact that you could not make up your mind on whether you wanted to be a scientist known for his expertise in your beloved cichlids or known for his quantitative skills. I'm glad you eventually selected the latter to find that your cichlids did not need to be abandoned. Partly, it was also because you needed to learn how to do things a bit less dirty and therefore also less quick. I admit I sometimes despaired over another bug that you had found, which you then told me about with a smile. But you grew up. I believe that the turning point was in 2013. You really, really wanted to go to Lake Tanganyika to dive for cichlids. We agreed on this, provided that you would hand in your first chapter before this. You lived up to this promise, but it did even more. You acquired funding for this trip from the KNAW Ecology Fund and the Gratema Foundation, not only for yourself, but also for your companion fish lover, Adriana. You contacted Walter Salzberger, and the 2 of you were allowed to join his crew in the expedition of 2013. It led to your most independent thesis chapter. Regardless of what the review process will do to this chapter, this was a great accomplishment. So yes, I am proud of you, but even more importantly, I enjoyed supervising you. Despite your nonchalance, you do have a sharp mind and a good sense of humor. For example, when I discussed with you the new rule that at the PhD defense, you have to promise to adhere to the rules on scientific integrity, we'll get to that later, you were quick to joke, so this means that this does not apply to the PhD thesis. I could go on and talk about how you managed to distract me with yet another humorous t-shirt. The fun times we had in Montpellier with Frans, Saleta and Julia, the making of the neutralizer that has become a standard tool in my teaching, your frequent statement that you would certainly hand in your thesis at the end of April 2014, even a week before that deadline, where not a single chapter was finished yet, and the stress you experienced at the very end of your PhD trajectory, where I kept pushing you not to lower your standards. But I won't go on. I just want to thank you for the pleasant road we traveled together and wish you all the best in your future career, which you are continuing as a postdoc in Leuven in Germany. So I would like to invite you to all stand up. Okay. By virtue of the powers vested in us by Dutch law, in accordance with the decision of the College of Deans of this university, I, Rampal Etienne, confer on you, Thijs Janzen, the title of doctor and all the rights associated by Dutch law or custom with a PhD degree at the University of Groningen. Do you, as a doctor, promise to act in accordance with the Netherlands Code of Conduct for Scientific Practice? You can be seated. Now that I have performed the task assigned to me, let me be the first to congratulate you on your PhD degree and to address you as doctor. So congratulations, Dr. Janzen.
Thank you.
As proof of this PhD degree, I will present you with this certificate, signed by the Rector Magnificus, Secretary and Supervisor, and sealed with the Great Seal of this university. But before doing so, very learned sir, it's my pleasant duty to congratulate you also on behalf of the College of Deans of this university on the degree you have been awarded. And I now declare this ceremony for closed.